welcome to my project on breast cancer subtype classification using machine learning and DNA mutation data. Breast cancer affects 2.1 million women globally every year. While advancement in treatment have improved outcome, early detection and accurate classification of cancer subtype remain critical challenges. The metabolic dataset provides a wealth of mutation level genetic data that holds the potential to reveal critical patterns associated with cancer subtypes. However, the high dimensionality and complexity of this data makes it challenging to extract actionable insights using traditional methods. In this project, we harness the power of machine learning to analyze genetic mutation data and predict breast cancer subtypes with high accuracy. Coming on to the dataset description, the dataset used in this project is sourced from Metabolic Study, which is publicly available on Kaggle. It contains two components. One is clinical attribute and another is mutation data. Clinical attribute consists of patient level features such as tumor size, patient ID, age of diagnosis, and the mutation data consists of the gene mutation profile for 498 genes or else for 498 columns with the breast cancer progression. So for this project, we have used cancer type detail as the target variable, and we are only moving forward with the mutation data for our classification. So moving forward with the code run. So I will be going through the code and not running it immediately because it would take long time to run it over here. So this is the import for the warnings and also the code attempt to suppress each and every warnings to create a clean output. Then we have done all the imports at the start of the code itself. Then we have loaded the data set into the cancer data frame. Then we have created two variables. One is clinical columns and other is mutation columns. So the clinical columns contains all the clinical level data set, and as you can see, we have avoided a patient ID for each of the subtype. Then we are checking if the rows contains any duplicates, and there is no such. Then we are checking if there are any null values in any columns present, and only displaying the columns which has none values. Then we are using simple imputer for imputing the missing values. For categorical data, we are doing the most frequent ones for categorical data set, and for the numerical we are doing the median for with the simple imputer. After doing all the imputation, we are checking again if there are any null values, and after that there were no null values present. Then we are performing one hot encoding for categorical data set, categorical data for each like clinical features as well as the mutation data as well. And after that we are combining both of the data frame into a single data frame, which is process data, and again we are storing it into a new CSV file, which is process data.csv. After that we are checking the variable the target variable subtypes, and there are five of these type. Then we are doing the level encoding for this so that every subtype is present in just one column, and that would be easier to take as a target column rather than using the one hot encoding method. Then we are using different methods to select the most suitable columns for our prediction model. So for mutation data as well as the clinical data, the methods are same. First we are using mutual information score to get the important features, and we are selecting the top 20 out of this. Then we are doing the random forest feature importance, and again we are selecting the top 20 out of this, and we are doing the RFE method and again selecting the top 20 out of this. Along with that, we are also taking the MI scores which are greater than which have an MI score of greater than 0.01 also, along with the top 20 of the MI score, and union all the features, that is the MI score greater than 0.01, the MI score which are top 20, then the RFE score greater than the RFE score top 20, and the random forest feature importance top 20, and we are doing the union of all of them. And then final feature count is now 185, which we are considering further for our prediction model. Along with this, we have done all these steps for clinical data as well, though we haven't used it in our prediction model there. But earlier the plan was to use both of them, so I have performed these steps for clinical data as well. After that, we have I have done the correlation checking as well with the target variable. The correlation checking with the mutation data is done with the target variable, and the perfect correlation of 1.0 was to be removed according to this logic. But there was no such, so none of the columns were removed, but this is an important step to be performed as well. After performing all of them, we have also done the check the level counts for each type for the target variable, and we have figured out that there is a heavy class imbalance, as we can see, and the metaplastic breast cancer has just one sample and it would create problem. So we have removed this class, and we have only done the prediction with these classes only. So these classes are highly imbalanced, so we had planned to balance these classes. Before that, we have removed the outliers with the isolation forest method with a contamination of 5%, which is used for clinical diagnostics purpose. 5% is used for that, so we use that. And then we have handled the class imbalance with the SMOT technique. And after that, we have displayed the training set size and the validation set size and the test set size. So to be sure that this is the correct one, I am validating that 1431 is the count of each classes, so into 5 is around 7000 something, so this is a valid count. No need to worry about this. Then we have scaled the data so that it doesn't create any issue for any traditional models. Then we have done the hyperparameter grid for each model we have tested for. So first we have done the logistic regression model. Then we have displayed the performance on each class, as well as the best parameters which was selected, and the accuracy of this. And this step is done for all the followed by models as well. The KNN, and we can see it is having also the similar output, the SVM, and this has also the similar output, the random forest classifier, and this has also the similar output, and the XGBoost model, and this has also the similar output. Now we have used a function to handle the display in a tabular form, so that it is easy to read by any of the user. We have written this function, and passing these parameters easily appends the model to the table, and the final look of the table which all we have tasted is comprised in a tabular form, so that it is easy to read and to compare as well. Now we are going to test the data set in the neural network model as well. So for that, we have done the one hot encoding of the target variable, and we have done the hyperparameter tuning for this as well, with the help of the function over here. And then the best model is selected, and on that it is trained and tested. And then we have also appended the results of the neural network in the final table as well. And again, for LSTM we have reshaped, which is apt for LSTM, reshape the model which is apt for LSTM. And then we have done the hyperparameter tuning, and hyperparameter after doing the hyperparameter tuning, we have done the similar steps for as all models. We have done the similar steps for this model as well, the LSTM. And then we have appended the result of this into the final table. And after looking at the table, it is very clear that among all the model, SVM performed the best, and among the traditional model, SVM performed the best, and among the advanced model, LSTM performed the best. And for each model, the confusion matrix was plotted, as well as the ROC AUC curve was also plotted. So looking at the confusion matrix as well, the true values are maximum in SVM and the LSTM model. And for ROC AUC distribution of the classes of the AUC curve is also more for each class in LSTM and SVM. So continuing with the slide part of ours, which is the key insights drawn from this project, which is the machine learning model successfully leveraged genetic mutation features for breast cancer subtype classification. And as I told earlier as well, among the advanced model, LSTM performed well, and among the traditional model, SVM exhibited the highest performance. Moving forward, the challenges faced during this project was class imbalance, as I told, that how we handled it with the SMART technique, and the high dimensionality of the features. So there were various techniques performed to select the important features out of this. And as I told, to avoid overfitting, we have included only the mutation data and avoided the clinical data. For future improvements, we are planning to combine the mutation data with clinical features using regularization to avoid overfitting. And use of raw DNA sequences can also be a future improvement, because exploration with those sequence based approaches such as k-mers and DNA embeddings for improved feature extraction could be a great work to be done in the future. And to the conclusion of this project demonstrated the effectiveness of the machine learning model in classifying breast cancer subtypes using genetic mutation data. And I, as told earlier as well, LSTM and SVM achieved the best results. Thank you.